All right, I am here to introduce our new speaker, and I just want to piggyback off Kim's talk. Also, ABRF is amazing, and I'm happy to explain anything because I'm a ABRF fangirl. So, all right. So our next speaker is Dagna, and she is the director of the University of Wisconsin Carbon Cancer Center Flow Cytometry Lab. She has been working in and managing and directing flow cytometry shared resource laboratories since 2000. Both at UW-Madison and at the University of Western Ontario. She is a certified cytometrist and actively involved in the Great Lakes International Imaging and Flow Cytometry Association, in the International Society for the Advancement of Cytometry, ABRF also, and she serves on the board of Cytometry Educational Associates as president. She's been involved in organizing, hosting, and teaching in the annual course in cytometry since 2019. And she's also served as interim director of campus research cores at UW-Madison from March 2020 to December 2021. Dagna is here to, her talk is called Let's Talk About Cores. So let's welcome Dagna.
Well, good morning, everyone, and thank you for inviting me. But before we get started, I want to do a little bit of audience participation. Don't worry, I'm not going to make you do groups or have activities. I just want you to raise your hand. So who here works in a core? Look at that. Keep your hands up if you manage a core. Keep your hands up if you're a core director. All right. Do we have any faculty supervisors or leaders or advisors for the course here? A few, that's fantastic. What about core administrators? That. Okay, and then I've got one more category. Who works in academic research, so university or a college? How about private research institutes? Couple. And anybody who works in a core in industry? They do exist, believe me. So that's fantastic. This is your network. This is your community. And I forgot to ask how to advance the slides.
There you go.
Hooray! All right, so here's my outline for today. I'm going to give you a little bit of context. How did I end up here? Why am I the one talking to you this morning? We'll talk a little bit about cores, and I kind of divided that into 4 categories. So staffing, instrumentation, fiscal/budget, education and outreach. And then we'll kind of talk a little bit about where we've been, what we've learned, and where we're going to go. But before I get started, I want to give a little disclaimer. Everything I'm going to talk about here is my personal perspective. So having worked in cores for the last 25 years, and the conversations, interactions, and collaborations that I've had with core staff here at UW, the researchers that use my core, the core administrators, and as well as other institutions and companies and groups like this. So why me? Molly kind of talked a little bit about this. I started out working at the Wisconsin Primate Research Immunology Services Corps when I graduated from college. I didn't really know what I wanted to do, so I was like, well, I'll just get a tech job for a couple years or a year, and then maybe I'll get a PhD, or maybe I'll go to med school, or maybe I'll go to vet school. And I kind of fell into this flow cytometry thing, and I was like, oh, I'm not going back to school now. So after that, I moved to the University of Western Ontario at the Robarts Research Institute, which is a private research institute in London, Ontario, and I ran a core there for about 3 years, and that was a one-person core. So I've worked in big cores, and I've done the solo thing, so I've got some experience there. And then finally I came back to UW as an instrument tech in the flow core here. And then when the existing manager retired, I applied for it, and I guess the powers that be decided that I could do it, and I've been doing it ever since. And then also, as Molly mentioned, I served as the interim director for campus research cores here on campus for a little less than a year. I've been involved in a lot of societies over the years. I would say there's no way I would be here without those societies. That's where I learned how to run core. So in short, the answer to why me is I've been around a while and I've seen some things. So let's start out talking a little bit about cores. So cores here at the UW. This is a map from our Office of Campus Research Cores website. And I like this one because it kind of tells you where everything is and kind of where the clusters of cores are. So the pink star is where we are. My core is, our main core is out where the big red 85 is, out on the west end. Some of you toured it on Wednesday. And then we also have a satellite facility where the red circle with the number 20 is in it. So we've got one sorter and one analysis instrument over there. And we partnered with the Biotechnology Center to put our satellite facility in there so that we can literally walk sorted cells down the hall to the Gene Expression Center for downstream single-cell genomics. That also allows us to serve more of the campus. It's not as big of a, you don't have to travel a mile to do flow cytometry. The structure of the Cancer Center, so my core is within the Cancer Center. We're a true Cancer Center core supported by the Cancer Center. So, and then we've got kind of three types or groups of cores. We kind of put them in different buckets. So we've got the Cancer Center shared resources. We've got umbrella resources where there are groups of cores that kind of work together. And then we've got partner shared resources. So maybe they're supported mostly by the Biotech Center and a little bit by the Cancer Center or other departments and groups on campus. A little bit about my core specifically. We have 6 full-time cytometrists, including myself, and 1 undergraduate. We have 8, I'll say 8.5 analysis cytometers. The half is we partnered with a department to help them support a cytometer that was critical to a group of 3 researchers. So they pay the service contract, they pay for the consumables, but then we provide general maintenance, QC, rigor and reproducibility. We calendar the instrument for them. And so we get a little bit of revenue from that additional support that we provide. So we have a memorandum of usage between us and that specific department. We have 3 cell sorters. We service about 200 research labs on campus every year, which works out to about 500 individual researchers or individual users. I would say we probably have maybe 40 to 50 consistent users. So that's users that are using it on a monthly, weekly, daily basis. On and off, we've served about 15 of the local biotechnology companies in Madison. We have about a $1.3 million annual budget just for our core. And I would say our cost recovery runs somewhere around 70% through user fees. And we have about 10,000 instrument hours per year. But that's just instrument hours. So that doesn't include like training and education or consultation or, yeah, I guess those would be the other ones. So let's move on to my view of cores. So over the years, I can distill it down to basically 2 statements. Cores are research infrastructure. And cores are cost-effective centers of excellence. So, and I think, I'm kind of preaching to the choir here. I think you all know this. But I think these are two statements that you could really keep in the back of your mind when you're communicating with administration about how your core is run, when you're working out your budget, when you're thinking about what kind of investments need to be made in the core. I think kind of keeping this in the back of your mind is going to help you with how you present yourself and the kind of conversations that you have with administration. All right. So let's go on to staffing. What do we do really well? Our staff has unique expertise. They're highly specialized. They're almost to a fault curious, creative, they're collaborative, they're fantastic problem solvers. They have a genuine concern for good outcomes. We're all really good teachers. And finally, we're generous with our time. I have yet to meet someone who works in a core that has an easy time saying no. And so there's a lot of people in here who are exactly like that. What kind of opportunities do we have to improve? We can work on recruitment and retention. Continued education and training for our staff, recognition, career path, and balancing the workload. So several years ago? A couple years ago at ISAC, there was a presentation by a woman named, I think her name was Kelly Veer, and she is from England. And she had this whole talk about how core staff are highly skilled scientists. And she titled her talk, "Not Just a Tech." And I hate that phrase. I don't ever want to hear that again. I don't ever want to hear anybody say, "I'm just a tech." I don't want anybody to ever say that about anybody who works in a core, because we're not just techs. We are highly skilled scientists. Instrumentation. What do cores do really well? We provide access to a fleet of instruments. Often this is instrumentation that a researcher would not be able to purchase, support, or maintain in their own space. We provide both cutting edge and basic instrumentation. We function with economies of scale. We're really good at quality control and maintenance, and rigor and reproducibility. What could be better? Rogue instrumentation. That does cut into a core's ability to operate. It's hard to do. A lot of researchers get instrumentation in startup packages. But what I would like to see is more, when these startup packages happen, or when researchers are thinking about buying their own, that they, at the very least, communicate with the core about what is this going to cost? And not just dollar amounts. What kind of staff time is it going to take to run the QC every day? How often do you have to do the maintenance? What are going to be your consumable costs? And maybe sit down and talk to someone who works in a core for that type of instrumentation. Tell them about your research plans and let them give you a realistic view of how much you're actually going to use this instrument. Because a lot of these instruments don't work well if they're not run a lot. And I think that we could really form some more partnerships with more of our researchers and departments and schools if we started having those kinds of conversations before the PO goes out. We're having a hard time maintaining the cutting edge. The time to instrument obsolescence is decreasing, and the cost of newer, faster, better instrumentation is going up for several reasons. And then, of course, right to repair. There's a lot of stuff that we can't fix, even if we know how, because we void the service contract or we void the warranty. All right, so on to the fun stuff, the fiscal. What's going really well? I would say we've got great resources and examples for rate setting. There's a lot of institutions that have some really good templates for rate setting. There's a lot of cores that kind of lead the way on this. Some of them have it available on their websites. You don't even have to talk to anybody, but it's better if you do. This rate setting it's a data-driven approach. We're looking at instrument use. We're looking at staff time. We're figuring out what are billable hours, what are not billable hours. Again, we've got economies of scale. And if you look at cores, if you look at their operating budget versus total research dollars served, we're doing great. What could be better? Institutional support. Isabel Gerard from the University of California, Davis, she actually was our previous director of campus research cores and why I was the interim. She moved to UC Davis, but she talks about a portfolio of support. So, and however that works out, you know, percentages are going to be different from institution to institution depending on what their funding is, but you've got user fees. You've got what I like to call and would like to see more often, true subsidies. So if you run a subsidy for a specific department or a center or a group, every month you should send a bill to them to cover the subsidy. So if a department gets 15% less than your flat rate, send them out a bill to fill up that 15%. Salary support. And then you've got campus funds, department funds, and hopefully we start seeing some more philanthropy funds funnel down into the cores. Philanthropy, we actually had some computer upgrades funded by philanthropy. We worked with our core management at the Cancer Center and our philanthropy group, and they actually put up a little website and people put donations in, and I could go on there and I could see, oh my gosh, this person gave us $1,000 towards our $40,000 computer upgrade bill. So that was kind of exciting. I think you've heard a lot about how external user contracts are complicated in academic situations. Maybe we start to think about bigger ways to do this, maybe umbrella ways, rather than individual contracts with individual cores. I don't know how that works. I'm not a fiscal administrator, but I'm willing to talk to anybody and help work those things out. Bottom line versus value added. I would like to have some conversations about just looking at the bottom line. I think we need to expand our metrics for core success. And finally, a little thought experiment. Are we overspending or are we underfunded? Probably a little bit of both, to be honest. And then education and outreach. This is really important to me. My course spends a lot of time on education and outreach. And I know that there are a lot of other cores and a lot of other technologies that are fantastic at education. Cores right now are training the next generation of principal investigators. We've got established training programs. They're virtual, they're in person, they're hands-on. We've got tech notes and SOPs and little how-to videos on our websites. We train them in instrumentation operation, experimental design, and optimization, a lot more and more on rigor and reproducibility. We do teach scientific theory. I know anybody who's ever worked in a course spent a lot of time talking about the proper controls over and over and over. And we support data analysis. So now we're starting to get into computational analysis and AI And maybe we need to hire computational experts to work in the cores. Or, maybe cores support 50% of one. Or cores share these types of positions. As evidenced here, we've got a lot of participation in regional, national, and international societies. A lot of these societies have specific core tracks, like GLIFGA that I'm a part of for cytometry and ISAC. And then finally for us, what I like to say, I think one of the reasons that we're a recognized core nationally is because we're involved in the annual course in flow cytometry. What could be better? I would love to see the ability to put in more protected time for my staff for teaching and taking webinars, seminars, and classes so that they can maintain their continuing education credits, so they can maintain their cytometry certification. I would like to see more recognition of core contribution to student and scientist training and education, because we spend a lot of time training people. So now I'm going to change gears a little bit and talk about kind of where we've been. And this idea came to me several years ago. I think it was, I don't know, maybe it was Nova or something on PBS, but they basically did a show on this book called Super Tall. And the thing that caught me was functional resiliency. How much time does it take to become fully functional after a catastrophic event? And I started thinking about what's my functional resiliency in my core. And here it is. So this is all of my instrument usage from October of 2016 through July of 2025. You can see where my catastrophe is, and you can see how resilient we are. So this is kind of broken down by different instrument type. Green instruments are kind of our workhorse cytometers. The pink ones are our spectral cytometers. The purple ones are our legacy traditional flow cytometers, and then the blue ones are our sorters. And even with COVID, I was really worried that we would have like pent-up usage, and we would have this like spike, and then it would go back down. And it doesn't really do that. You can see our trend line is just increasing. But my data set only goes till the end of July 2025. I think I'm okay through the end of September, but I think I'm going to start seeing things in October because there aren't as many postdoc positions as there were last year. So I have less customers. So now I have to think about how am I going to weather this? So I think of it in terms of like a continuity of operations plan. So I think all of the research labs on our campus have to come up with this continuity of operations plan. Like, what if there's a fire? What if there's a tornado? If you're on the West Coast, what if there's an earthquake? But what I'm starting to think about is how to rethink and expand this for a slow-moving catastrophe. And some of the things that I've started thinking about is how do they do this in resource-poor areas? What kinds of programs and support do they have? So, for instance, there's a group, and I should know what it is, but I don't remember it. But there's a group of folks within ISAC, the International Society for the Advancement of Cytometry, where they purchase and refurbish what we would consider obsolete cytometers. And then they're able to ship those cytometers to other countries that don't have they don't have research institutes like we do. The other thing I'm thinking about is, are there tried and true methods that could be revived? And I think there's a lot of scientific equipment, and we all have it on our campus, We've all got centrifuges that are 40 years old and they're still running. Are there other types of equipment like that, or are there other types of assays or metrics that we could do cheaply and fast enough? Like, what kind of economies can we get from that? So, kind of think about a roadmap to recovery so that you're ready to rebuild when the pendulum swings the other way. Because what's going on right now is absolutely not sustainable. This won't last. It never does. It's just, how long is it going to last? And then I've been thinking about new solutions to old problems. And I heard this story while I was driving home the other day, literally just a few days ago. And it was a story about the Houston Rockets, and their starting lineup averages 7 feet. And it's potentially the tallest starting lineup the NBA has ever seen. And it's unique because it hasn't been done before. I mean, there've been a few teams here and there that have done it because, I don't know, people were out or it just happened. But the old way of thinking was that tall players would stand by the basket, they would dunk, they would post up, and then Kevin Durant shows up, and he's a fantastic shooter, he's an excellent ball handler, and now the Houston Rockets have the tallest starting lineup. Are there things that we just haven't thought about yet? So start thinking outside the box, looking in other areas, and maybe you'll come up with an idea that works for your core. So again, those new solutions to old problems, cores are research infrastructure. How does that change how you run a core if you think about it as research infrastructure? How does that change if the administration recognizes cores as research infrastructure? Does that mean that we're eligible for F&A? Maybe it does, maybe it doesn't, but why not ask the question? And again, how are we cost effective? How can we be more cost effective? So some of the hard questions that have been rolling around in my mind are, should we be rethinking or maybe discussing the PI-centric approach to research? What if we flipped it? What if we did the thought experiment and we flipped it and we put cores at the top? And we made them into these centers of excellence. And researchers didn't have to worry about spending money on their research grants for instrumentation because it was already there. The training is there. The technology is there. The specialization is there. Remember team science? We talked a lot about it. Let's put cores on the team in team science. Let's have a really tall starting lineup. Stop rogue instrumentation. We can do this. When I talk about it, I'm told, well, we can't do anything about it. They say they need it. And I'm like, no, we can do it. UPenn does it. You cannot buy a flow cytometer on that campus unless it goes in their core. They've got 40 instruments, over 5 or 6 sites on campus, and they control all of it. A question to ask administration, are you going to subsidize? And I think I'm going to stop using subsidize and start saying invest. Are you going to invest in your own cores? Or are we going to continue to allow researchers to subsidize cores at other institutions and private CROs? Because every research dollar that goes out of your university goes out of your university. It goes to somebody else. You want to keep it at your university. Maybe a collaborative approach versus a top-down approach to core sustainability. Let's start having real conversations with the people who work in the cores, rather than some administrator coming to you and saying, well, you're at 70% recharge, I need you to be at 100. And that's it, no discussion. Let's really think about it, and think about the return on investment versus the bottom line. What do you get for that investment? Yeah, maybe you put $250,000 a year every year into a flow core, but what do you get for that? What are you buying? Don't think of it as filling a hole or backfilling or subsidizing. Think about it as investing and buying something for your researchers. And I think all of this is possible by building community. And one of the things that I would really like to see, and I think organizations like ABRF and MWACD and some of the flow cytometry groups are really in a good position to do this. Let's have some comprehensive and robust core benchmarking. Can anybody tell me what the average cost recovery is for cores in the United States? You can't, because the data doesn't exist. We don't talk about money openly. What's the average portfolio of support? You know, who's got a lot of philanthropy? Once you know that, you can talk to them and you can figure out how they do it. You know, who uses a large percentage of their cancer center support grant for the cores? Figure it out, find out, talk to them, figure out how they do it. Appropriate salary comparison data sets. We got to go outside of our own institution, and we have to really think about the individual technologies. Because some technologies, it's not fair. Ideally, everybody would get paid the same thing and we'd all be comfortable and happy. But realistically, that's not true. So are there specific technologies where those salaries should be higher because they're more in demand and they're more technical? And I think ABRF is doing a fantastic job of this. Wider consensus, adoption, and recognition of best practices. And I don't think that's just for the technologies and the techniques. I think that's for how we run cores. So, you know, collaborate across cores, across disciplines, between your cores and your research labs, between your cores and your administrators. One thing that, and I was so happy to hear it, because I've heard rumors of this, where researchers would like a core pipeline to the point where they don't have to touch anything. They come up with an idea, they come up with a research plan, and they talk to the cores, and they start out at the animal core. And the animal core hands off samples to another core. That core processes those samples, runs those samples, optimizes the protocols, does the data analysis, and they're just getting fed data from point to point to point to point. An academic CRO. How do we do that, and what do we need to do it? Let's start talking about it, because researchers are starting to ask for it. We want to share our experiences and data. If you tried something because you thought it was going to increase efficiency and it worked, tell everybody. If it didn't work, still tell everybody. Because no is a valid research answer. And I would argue that we get a lot more no's than we do yeses when we're doing research. If it was easy, I don't think we'd do it. What about external advisory boards for cores? So, for me as a flow core, maybe I could create an external advisory board of other core directors, faculty advisors, or maybe even senior staff from other institutions, so that I can leverage their processes and their experience to help improve my core. And then finally, let's talk to each other. Share your goals and your crazy ideas and your secret evil plans. And by evil, I mean good manipulative. So no one can do everything, but everyone can do something. And then my final thought, if you expect to see the results of your labor, you simply have not asked a big enough question. And when I think about this, I mean big in every way. I mean big in your scientific goals, big in your instrumentation goals, big in your staffing goals, big in your timelines. What is your legacy going to be, and how are you going to hand that off to the next generation? We don't think as far ahead as we should. There are Indigenous communities that think 7 generations ahead. Every decision they make, they think, how is this going to affect my descendants 7 generations down the road? And I think we can start doing that. So finally, my thank you slide. I have absolutely not listed everyone who's helped me. The kind of list of names at the bottom are the people that I spend a lot of time talking about cores with. But it takes a village. So I'll take any questions or discussions or whatever, and I'm available till the end of the conference today if you want to grab me and talk to me.
Any questions? Ooh, way over there.
It's okay, I have my running shoes today.
Great talk, Dagna. How do you cover the costs of training? I mean, I think that is a real thing for all of us here at the UW and I'm sure other places, but with being fee-for-service, I guess I'm curious to see how you help, cover that cost or others do.
Yeah, so I actually build it into my budget. And then I think really carefully about what is it, I want to get the biggest bang for my buck. So everybody who works through me gets to go to the annual course at least once. And that's been true since before I started working in the Corps. But I've been really lucky to have a really stable and fantastic team. So that kind of saved on costs. But, I think about things like what meetings have really good core tracks. So where are they going to get to meet and talk with colleagues from other institutions? Local meetings, things like GLIFCA, the Great Lakes Flow Cytometry and Imaging group. This meeting, where can you send them locally if they can't go everywhere? And then, of course, are there virtual options? And if there are virtual options, what I like to do is to have maybe a group of 2 or 3 just book a conference room and do it there. So you're not by yourself. You're still having an experience with another person. You've got someone to talk to and communicate and kind of think out loud about these crazy ideas or new things or things you want to try. So it's always a challenge, but build it into the budget. Get it approved by your administration. Yeah.
Hi, thanks for the talk. It was fantastic.
Thank you.
And your journey through cores is very impressive and inspiring.
Thank you.
And one bullet that you brought up that hopefully you can expand upon is the retention and recruitment piece of that for people that are in your early stage of your career. How good are we doing as a community in building that group of young, excited, technical scientists that want to move into the pathway that you've shown us?
Yes. So that was another thing that Kelly Veer brought up, and one of the reasons that she really kind of embarked on this this journey that she had, where she actually put together a group in the UK to specifically recognize and support technicians. And part of the reason that she was so successful with that is because they didn't have a good pipeline for, you know, they didn't have a good pipeline at all for, you know, starting out in the field. So I think there's some kind of low-hanging fruit. You could do, I mean, if you're thinking about, you know, next generation or how do you get new people in, do some outreach. Participate in core tours. If your university has, here we have something called science expeditions. So we've participated in tours for those. So that brings in kids and their parents from all over southern Wisconsin, can come in and see what's going on at the university. You know, if you've got kids in school, like parent-teacher days, and, you know, bring your parent to school day, and, you know, do what you can to inspire people about science. And then I think in terms of like actually retaining who you have, I think it's you know, you have to strike a really good balance. Because there's quite a few people on this list that used to work in my core, and I mentored every single one of them right out of my core. So, but it's benefited my core, because now I have collaborators in the companies that make the instruments, that make the reagents. You know, I've got technical application specialists that used to work in my core. The Cancer Center Administration has someone who used to work in my core, which is fantastic. So, you know, and the other thing too is that I think when you do that, you see that it encourages the staff that's still there, because they see how you as the leader are willing to support people, whatever their next move may be.
I have a question from our virtual audience. Carrie Schrey asks, in your evil plans, can you give an example of how you add goals for others to achieve the plan?
Yes, and I'm gonna pick on Alex. So Alex Henkel used to work in my core, and I noticed pretty quickly that he liked the business side of it. So when immediately, as soon as I realized that, I was like, great. Do a rates analysis for me. Oh, I'm sorry. I said, you know, as soon as I realized that he was interested in that stuff, I handed him the rates analysis, and he ran with it. To the point that while he was still working in my core, he was working with the CFO of the Cancer Center to set up a template. And then when a position opened up in that core administration space, he applied, and of course he got it, because he was able to build those kinds of connections. And from, you know, the bonus to me, the secret evil plan of it, I want more administrative support for the course. And I got it. And Alex got a job he likes. So win-win.
I would just add, in terms of the next generation of core staff, is that if you have the ability to hire student hourly workers, that is a great opportunity to expose people to this type of work that maybe hadn't thought about it before.
Yeah, I would agree with that. We had a student worker, and we usually offer them more hours over the summer. And then we'll train them like on sorting or maybe helping out with more of the assays and the bench work. And we had one several years ago who ended up working for us after they graduated for 2 years. So that could be your pipeline, and it worked well for us. So thank you for bringing that up.
I actually have one,
Yeah.
and I have the microphone. In your instrumentation, when you were talking about investigators getting packaged, to buy instruments, and it may be for the whole room, not necessarily you, Is there a way that instead of getting them to buy an instrument that is already in a core, or that they can maybe get vouchers to work with cores?
I think that's a great idea.
Because that helps them figure out starting their lab, seeing what it really means to have an instrument.
Yes.
And that could be a way to get them to know the cores also.
Yeah, and I think it's a great opportunity for the cores because that may be an opportunity to bring in several instruments and do instrument demos.
May
And-
not necessarily, I didn't mean to interrupt.
No.
May not necessarily come from, the cores giving the vouchers. They can work with the institution that instead of giving money to buy an instrument that is going to be not as used, then they can just give money to tell them to go use the cores.
Yeah, I think
Research infrastructure.
Yep, I think that's a great idea, and I think something that also follows along those kinds of lines is, the kind of trickle down idea that if a PI, their research project turns out to be more core use than it was, there's a lot of bridge funding that goes to many investigators. But what if you track that money and you figured out how many times do you do that as an institution? And wouldn't it just be better to say, all right, we're handing out $500,000 to researchers to use this core. Why don't we invest in the core and bring the rates down for everybody?